just like to welcome everyone to our Prepit CEO Corey joint rounds today. Mark Phillips, who's a PhD candidate with our team at McMaster, is going to talk about meta-analyses and meta-analyses part 2. I think Kim did a wonderful job with part 1 a couple months ago, so hopefully this will build on it.
Great. So thanks a lot. Thanks for having me, everyone. As Sheila said, I've been a PhD candidate with the group, and before that was in my master's and started in my undergrad actually with a research project. So I've been with the team for, I believe, coming up on about 8 years. My PhD is primarily focused around meta-analyses, in particular network meta-analyses, and improving reporting and interpretation of network meta-analyses, and we'll touch on that a little bit in the second half. To start, we'll just, I won't get into a ton of detail about what a pairwise meta-analyses are. Obviously, Kim had a great presentation around, the basics, but again, just as a quick refresher, obviously, most people know when we're talking about a pairwise meta-analysis, we're really looking at pooling all of the available studies, typically trials, for one that all have a common comparison, and pooling them to find a pooled estimate across all of those studies. This is, again, typically done, there's a number of kind of justifications and pros. Obviously, we're increasing our power and our precision from single studies by being able to amalgamate all of that data. It's quality evidence from a large sample size when all of the studies are pulled together, and probably not more importantly, but something that's often overlooked is the actual power for subgroup analyses. Trials almost always, I know, this group in particular goes beyond this, but typically trials are powered for their primary outcome, and that leaves subgroup analyses almost always underpowered within an individual study. And our ability to pool study data and then potentially do subgroup analyses within a meta-analysis gives us adequate power to maybe understand some of those subgroups in a way that I would say, the vast majority of trials can't do. I know this group had, sometimes with our sample size calculations does focus on powering for some of those subgroups, but it often is not done that way. And the cons, I stole this from Kim because it's very true, is sometimes people believe because meta-analysis, depending where you look, is sort of the top of the evidence hierarchy. It's not a really accurate representation. A meta-analysis is really as good as what you're putting into it. So if you put in a bunch of very poorly done trials, your meta-analysis will be, poor. It doesn't really change, what is going into it. So that's something to consider as well, and not thinking that just because we've pooled a bunch of data, somehow it's, better data. It's only as good as what we're putting in. As I kind of alluded to about subgroup effects, there's been research into this, actually demonstrating, subgroup effects in randomized trials. The conclusion here being, oftentimes claims were strong around subgroups, but very often there was they were underpowered and not credible subgroup analyses. So to my point of the importance of pooling data in a meta-analyses, it's almost the most beneficial in this regard in increasing our power specific to looking at subgroups, as well as just say the primary outcome itself. In terms of guidance around conducting meta-analyses, again, Kim touched on a lot of this, so I won't go into detail about these, but there's a few important items. PRISMA is the reporting checklist that is, I guess, the standard for meta-analyses. So when doing a meta-analysis, it's probably a great place to start in designing your meta-analysis and going through the checklist and making sure, everything that's in the checklist you've incorporated. There's also Prospero, which is the registry for protocols for reviews. There's an increased push to have reviews also have protocols associated with them, for obvious reasons. But this is another system that highlights specific categories in which you're to fill out within your protocol. So it starts getting you thinking about all of the information that you need to be thinking about. And then there's all kinds of other guidance out there. BMJ has a best practices section on their site, and a lot of say editorial type documents around all of the aspects of conducting a meta-analysis. So we won't go into that again because Kim did, but there's a lot out there to kind of help guide conducting a meta-analysis. And what I'll talk about a lot is primarily to move on from, part one of this session is a lot more around statistical considerations. There's a lot of them, and a lot of them are sometimes confused or overlooked. And they're important to at least understand when you're reading a meta-analysis, but also very important if you're conducting one. The first, very quickly touching on fixed versus random effects models. This is something that generally speaking just isn't talked about, discussed, or justified in why one was used over the other. The long and short of it is there are almost no scenarios in which a fixed effect is really considered favorable. What I mean by fixed effects is a fixed effects model assumes there is no heterogeneity between the studies. So all of the studies are theoretically identical, and the only variability you're seeing is patient variability. And that just, is somewhat from a practical standpoint a bit nonsensical. All studies are going to be slightly different, and to really think that there is zero heterogeneity across studies and their designs is really not logical. And most people use a justification for when heterogeneity is very small and you have your I-squared value for your heterogeneity. If it's 0, then you should use a fixed effect. But if you do a random effects model in that scenario, it will actually just give you the exact same result as a fixed effect. So even in that case, there's no real reason to do so. And we'll see a little bit more around some guidance documents that really promote random effects being kind of the standard model that should be used. Certainly, I've not come across any real justifications for fixed models, and they often over exaggerate what the effect is relative to the random effects model. So the effect will often appear, larger in a fixed effects model. So it's the less conservative option. The other paper I wanted to draw everyone's attention to is a fairly new paper, and this is also relevant for randomized trials, and it's a great resource for trialists as well as meta-analyses. And this is called the Iceman credibility instrument. And this is the development of credibility assessment for effect modification, which would be subgroup analyses, for example. That's a broad term for any type of subgroup analysis, sensitivity analysis, determining if that subgroup is credible or not. And this is a great paper led by a number of individuals here at MAC who have put out this guidance. So it directly relates to meta-analyses. It also directly relates to randomized control trials when deciding what subgroups it should be analyzed within the analysis. And here's a snippet of their table that briefly summarizes this. So you can see the dashed out questions are ones that aren't relevant to randomized trials or meta-analyses in their respective columns. So the questions that you're to ask when you set up a subgroup are, is the analysis of the effect modification based on a comparison within the trials versus between? What this means is, do you have data on the actual within the trial of, say, elderly versus young patients, or are you categorizing studies based on the mean age in that study? That's one consideration. Obviously, to categorize and do a subgroup where you're analyzing, say, studies that had a mean age over the age of 65 versus studies that had a mean age below 65, there may have been many patients within those studies that actually fell into the other group. So it's not as precise as a subgroup, opposed to actually having the data from the study within the study to know the outcomes of the people that were below and above. Also looking at, were the effect modifications similar from trial to trial when you're doing this in a meta-analysis? So when you're looking at the age subgroups above and below 65, are the trials consistent in showing kind of the same results generally in those age subgroups? For between trials, is the number of trials large? I won't go through all of these, but you can see here there's 8 criteria for meta-analyses and 5 for randomized trials. That this paper gives the ability to kind of score each of these to determine how credible the subgroup is. So for someone reading a paper with a subgroup, this may be helpful, and also developing your subgroups for future trials or meta-analyses, also very important. And I will note, you can see here, did the authors use a random effects model is one of the questions, and that's where they are explicitly preferring a random effects model in this scenario, to say, if they use the fixed effects, that may be less credible, most likely. So at a very sort of quick activity, I was wondering if anybody, not so much an activity, but we'll stop here, and based on kind of discussing these subgroups and these criteria, does anyone, first of all, if everyone just kind of think about maybe a study, or whether it be a trial or a meta-analysis that you're currently a part of, or that you've recently read, or even just make one up, and can you walk through these core questions and really think about that subgroup, and if it meets these criteria or not? Obviously, for say a trial, I know obviously maybe Prep-It, for example, is something that some of you might want to think about and look at these questions and think, do the subgroups that we were thinking about meet these criteria? And if anybody has the courage to share their thoughts around a subgroup, and based on this, whether they think it would be credible, what could have been done maybe that would have been more credible, or something to consider for future subgroups. I'll give everybody a second just to kind of look through and think about this. So does anybody want to, I can provide an example, or does anyone want to talk about a subgroup that comes to mind that's maybe part of a current study, and how it may fit with these? What about, I know in Prepare, you're talking about subgroup analyses. I know it's analyzed in that study, or looked at in that study, around the amount of wound contamination, and that is, a subgroup that is being analyzed for sterilization in that study. So was the subgroup hypothesized a priori? Obviously, you wouldn't know if it's correct, but you know, is there a priori assumption around you know, what you may see in that subgroup of you know, more contaminated versus less contaminated injury? I'll assume yes. I mean, if anyone speak, I'm assuming.
Yeah, we stated all of that in the protocol, Mark, for the three subgroups that we're planning to do for the PrEP trials for APREP.
Right. So that's great. So that's been you know, that's been, you know, stated a priori and supported by prior evidence. Again, yes, right. You're supporting that by, you know, there's some biological, you know, clinical justification for why you believe that. Is there a test for interaction? This is something that would be specific to the statistical analyses. And is there a small number of effect modifiers or subgroups being analyzed? Sheila, I think you just mentioned 3. So this is important in thinking that the trial should not be a fishing expedition, as we sometimes say, in doing, you know, 50 subgroup analyses and hoping that you know, spot out one. So all of these, again, are probably criteria that inherently, you know, most of us are thinking about and just generally doing, but this tool is kind of a more formal way to reference and state, you know, we have based these subgroup analyses on some core criteria. Quickly talking about tools and actually conducting meta-analyses. I know Kim talked about this a little bit. For those of you at Mac, this may be a little bit Mac focused, but you know, it generally will apply elsewhere. This top section, this is the portal at the Hamilton Health Sciences website for the Health Science Library. And you can see, you know, conducting your search, you know, the start of actually conducting your meta-analyses after you've planned it out. You can do all of your searches here in the appropriate databases. And I know Kim walked through this in more detail. We use Covidence typically, again, or any other type of reference manager. And this can also be used for data collection. I'm still, I guess, in the somewhat old school use of Excel opposed to Covidence data collection. So I do collect my data in Excel. And then Review Manager, Cochrane Review Manager is a free software that is very intuitive, very easy to use. And there's all kinds of, you know, YouTube guidance and summaries on how to conduct the analyses in Cochrane. And then Grade Pro is another pretty important tool in using Grade, which we can talk about briefly, to actually assess the credibility or the quality of the evidence in a meta-analysis. And Grade Pro generates all the summaries and the tools. And with this kind of set, or, you know, replace maybe Covidence or Excel with others, this is generally the process. And again, I'll refer to Kim's slides where she really kind of talked through this more specifically. So talking about statistical analyses, there's, in a meta-analysis specifically, there's a few types of effect estimates that are used. And I want to just touch on them just to go over with everybody in case you're not familiar. When we're talking about continuous outcomes, for example, like function in this example, a continuous outcome would typically be reported obviously as a mean and a standard deviation, as you can see in these studies. And there's typically 2 options that this would be presented within the actual meta-analyses. This would either be a standardized mean difference or just the mean difference. What that means is, obviously the mean difference is simply, you can see for this first study, Keating, the mean in the THA group is 79.9, and the mean in the hemiarthroplasty group is 77.1. The mean difference is very logical. It's very simple. It is the difference between these two. So that's 2.8. But when you look over here, this is reported as a standardized mean difference. And you can see that they're reporting the standardized mean difference in this group is 0.18. So this is a different scale than what this was measured on within the study, what functional tool they used in the study. The reason this is done and the reason standardized mean differences are used is primarily to be able to pool results of an outcome like function despite the actual tool that or measurement tool that they used. So Keating could have used a WOMAC function. One of these other studies could have used, a VAS function, and they could have all used different actual tools. And those tools may have been scored differently. One may be, a score from 0 to 100. Others could be from 0 to 20. And in that case, they're pooled using a standardized mean difference, because the mean difference obviously doesn't make sense. They're not on the same scale. So everything has to be converted into this similar scale. And how that's done is the mean difference, as we said, it's 2.8 here, is divided by the standard deviation within the study. And not to get too, I guess, technical, that then puts all of these studies on the same scale of what we would call standard deviation units. The big issue with this is, what the heck does a standard deviation unit, refer to, or clinically? Like you can see here, there's a difference here that favors total hip arthroplasty with a standard mean difference of 0.29. Like, what the heck is 0.29 really referring to? And that's a big issue with standardized mean difference. And what typically is done or is promoted is to actually use tools, and there's Cochrane guidance, and I believe GRADE has some guidance, to convert this number back to the most common scale. So if that's WOMAC function, they would advise to take the standardized mean difference and convert it back onto that WOMAC function scale so that clinicians can make sense of it. Because the standardized mean difference is not very intuitive to interpret, but it does play a very valuable role in allowing you to pool all of the studies regardless of what tool they used. The next point around continuous outcomes in particular is this concept of a minimum important difference, or minimum clinically important difference, it's sometimes referred to. Statistical significance does not mean clinical importance. These are two entirely different concepts, right? So with statistical significance, the only things that really are determined with statistical significance are the estimate of the effect, the variance, as well as the sample size. So when we have a very large sample size, particularly when now we're looking at, kind of this new concept of big data analyses, you can have very small effect estimates that are statistically significant because of the massive sample size. So that effect estimate may not actually be clinically important. despite it being statistically significant. So a minimum important difference is a concept that tries to tie the importance to patients back to that estimate. And for a meta-analysis, this is very important to utilize a minimum important difference when interpreting the pooled estimate to decide if you pool a very large number of studies, again, your sample size might become very, very large. You're maybe more likely to observe a significant difference within your meta-analysis. But it really needs to be tied back to clinical importance and the clinical relevance. And that concept is the MID, and that's the smallest difference that patients would perceive as beneficial or harmful. There's all kinds of research into this, and I would advise whenever you're doing a meta-analysis and you've decided on your outcomes, say you're looking at a functional score, there's databases and a lot of publications where you can search and find, the Womack function scale and find what the MID is, and it may be, 8 points on the Womack function scale is considered meaningful to patients. Now, if we kind of changed from continuous outcomes to talking about binary outcomes, events, they're most commonly reported either as an odds ratio or a risk ratio. Many of you probably see this all the time, but oftentimes, there's a very nuanced difference between the odds ratio and the risks ratio. And typically, if you're pooling randomized trials, the risk ratio is one appropriate and two much more intuitive to interpret for clinicians, for readers. An odds ratio is a bit more abstract, I guess. Not a lot of people think in odds; they think in risk. So another thing to consider when you're developing your meta-analysis, if you're conducting it, how will you report the outcomes? Are you reporting them as your continuous outcomes as standardized mean difference because you have many different scales? Are you reporting it as a mean difference? Are you reporting your event outcomes as odds or as risk? These are all things, to lay out and decide and understand sort of the differences between them. Okay, so I have a quick exercise, and I don't know how or if I'm able to see this, but I have 3 scenarios, and I'll read them all out, and maybe in the chat, if everybody could just write the number 1, the number 2, or the number 3 to what you think is most appealing if you were a clinician in a particular field. There's been a novel implant just come out, and they had a study published. Which of these 3 results would be most compelling, or you'd be, I guess, most enthused by? So number 1, the novel implant had a revision rate that is 50% of the gold standard. So they had half as many revisions for the novel implant. Number 2 would be the novel implant's revision rate is 1% lower than the gold standard. And number 3 is for every 100 patients that were treated, the novel implant had 1 fewer revision. So I'll leave that up on the screen. Everybody can read. And if you just want to put in what you think is okay, so far a few ones. Kim has an ambiguous. She needs more details. I'll share those soon, Kim. A few more ones coming in. So generally, it looks like most people are saying 1, right? Having a revision rate that is 50% or half of the gold standards. That seems the most appealing. the most beneficial. But they're actually all potentially the same. And to Kim's point, there needs to be a bit more information there that was purposely withheld to be a little bit provocative. But for number one, you can imagine if the novel device is half of the gold standard, if that revision rate was 1% in the novel device and it was 2% in the gold standard, then the relative difference there is 50%, right? 1% is half of 2%. Now, when you look at option number two, the device revision rate was 1% less than the gold standard. Again, the same thing, when the revision rate is 1% less, and that's called the absolute risk reduction. And number three, the number needed to treat is a function of the absolute risk reduction, in which 100 patients needed to treat to observe one less revision is, sorry. Need a quick drink. That's also the same as saying that the absolute risk reduction is 1%. So these actually all say the same thing, and it's kind of a important consideration, one, particularly for meta-analyses and how meta-analysis results are reported, and two, when you're reading papers, particularly say industry papers, they're going to present their results as number one, right, and say, look at how great this is. And it's less enthusiastic to say they reduce the revision rate by 1%. It's much more enthusiastic to say, the risk was reduced by 50%. And the general guidance is that all of these should be reported, because the only way to get a complete picture of the results is to know all of this, is to know the relative reduction, but also what that really relates to in an absolute sense, because an absolute reduction is what clinically you might refer to when a clinician is thinking about how much safer is this device or not. You'd often think in an absolute opposed to a relative. So something to consider. And in meta-analyses, this is important, and grade explicitly, the grade when you conduct the grade assessment for a meta-analyses, their tables explicitly have 3 columns for each of these. So all 3 items are reported. So it's just kind of the new general practice. You can see here a quick example. So you can see the middle column there is the relative effect, and then anticipated absolute effects. They're talking about absolute effects, and they report a number needed to treat. So now I'll talk about network meta-analysis for the last little bit. Before getting into that, are there any sort of questions around, and it doesn't have to be anything, it could be outside of what I've just discussed with pairwise meta-analyses. Are there any particular questions around, statistical concepts or just concepts in general around meta-analyses that anybody wants me to touch on before we're moving on, or I'm open to any questions here. If not, we'll spend a little bit of time going into a bit more detail around network meta-analyses. A network meta-analysis is an extension beyond pairwise, where we can now compare any number of treatments, 3 or more. Obviously, in the vast majority of clinical scenarios, I think it's very rare for there to simply be 2 options. There's almost always more than that. So a network can give a holistic sort of summary of all of the different options and the evidence behind them. And you can see here a quick graph. This is over time, the number of network meta-analyses published in knee arthritis alone is going up exponentially, and presumably this is also happening across many other areas. So it's certainly kind of a novel hot topic. So why network meta-analysis? As I said, oftentimes there are many, many different treatment options for any given condition. So we can provide a lot of information in a single analysis to compare all of the different options. This is an example, not within orthopedics, but I like to share this one of, this is a study of antidepressants, and it really illustrates what a pretty extensive network may look like, where all of the different treatments are included here, and all of the lines represent trials comparing them, and the thicker lines simply meaning there's more trials, and a thin line being less, and the green circles represent how many patients are in those, each bubble. So this really illustrates the power of a network and the possibilities to really give a pretty complex overview of a clinical area. The main, I guess, benefit as well to a network meta-analysis, you can obviously, going back to this, conduct, in this case, a very large number of pairwise meta-analyses to compare all of these individually. That's certainly possible. But networks allow for indirect comparison where there's no trials published. So you can see here, if A and B have both been compared to placebo and there's no trials between A and B, we can actually draw some inference between A and B and provide some data around the perceived benefit. So if treatment A is 15 points better on a VAS scale than placebo, and treatment B is 10 points better, very sort of, this is a bit oversimplistic of how a network would calculate this, but generally speaking, we can say you would presume then that treatment A is about 5 points better than treatment B. That's really the overarching concept around an indirect comparison, and that's kind of how they are determined. Obviously, taking into account variability with standardized or standard deviations, but this is the concept. So we're allowed to actually compare things that have never been compared in direct trials. And apart from all of the concepts that apply to a pairwise meta-analysis generally apply to a network. There's a few kind of key topics that are specific to network meta-analyses, and everything else really, is transferable from, our knowledge of pairwise meta-analyses. And these three concepts that I'll talk about, and we'll walk through. One is heterogeneity, which is the same as pairwise meta-analyses. We're talking about discrepancies between the studies within a comparison. So, some studies say treatment A is favorable and some say treatment B is favorable. There's heterogeneity in that specific comparison. Specific to NMAs, there's a concept of inconsistency, or it's sometimes referred to as incoherence. And this is a discrepancy between the direct and the indirect estimates. And we'll get to this on the next slides, but essentially when a head-to-head trial has one result, but the indirect comparison we just talked about says the opposite, then there's inconsistency there. And then transitivity refers to an overarching assumption that there's discrepancies across the comparisons in the network. And this is more subjective in the eligibility of the trials and the eligibility of each studies in the comparisons, that across all of the different comparisons, they're similar. And we'll talk a bit about what that means. But generally speaking, all of the trials should really only differ in the treatments that were given, and they should be in the same populations, in the same circumstances, or else there can be, some pretty serious down analysis issues with transitivity. So going back to the direct and indirect comparisons for the network, a network effect is what we call the accumulation of the direct effect, which would be all of the results from the trials comparing those two, as well as the indirect effect, which, as we saw, is done through a common comparator. And then the network effect, the estimate that's given from a network meta-analysis, is called the network effect. And this is a composite of both of these. But what happens if the direct and the indirect don't agree? So we have a head-to-head trial, maybe saying that treatment A is better than treatment B. But when we do our loop through placebo, for example, and there's been trials comparing A and B to placebo, it actually says that treatment A is worse than B going through our indirect estimate. So this leaves our network a bit at odds because they're not consistent. And this is what's referred to as consistency, and it's a test that can be done within network meta-analysis statistical software to really cover comparing these and determining if they are consistent or not. So what do we do in this scenario is often the decision, and there's, for anything network meta-analysis, there's a lot of papers published by grade, by individuals within the grade group. Many of you probably know Romina. They have a pretty comprehensive catalog of publications around all of these concepts, if anybody really wants to get into it. But maybe I'll pose this as a question. What would we do when the direct evidence from, say, a trial or a number of trials says one thing and our indirect estimate is saying something completely different, or maybe even the opposite? Does anyone want to generally have their take on what we might consider doing in this scenario? Or what would you believe? Nobody's sure. So oftentimes people might say, you would believe what is actually from the actual trials, the direct evidence, right? Those are results from trials. And the indirect effect is kind of calculated or manufactured through a loop through, say, placebo. But it's not always as simple as just believe the direct data from trials, and you would favor that over the indirect, because it's possible that this direct effect is maybe 2 or 3 very poorly done trials, and the indirect effect is informed by a number of extremely well done, high quality trials that compared to placebo. So maybe treatment A has a whole bunch of very high quality trials to placebo, and treatment B has a number of very high quality studies against placebo. And then there's 2 very poor trials comparing the 2. In that scenario, and this entire paper by Romina really goes through when to make this decision of, are we going to believe the direct or the indirect for this comparison? And that's a scenario to think about. And in that scenario, we may actually defer to the indirect estimate as being informed by better evidence, and therefore it may be the more believable estimate. So something to certainly consider. And the last concept that it brought up is transitivity. So again, the only difference between all of the comparisons is the interventions given. And if there's systematic differences, transitivity may be invalid. It typically, I guess, presents itself as inconsistency. It's kind of this issue that is subjective in you're saying you need to have a general sense or a general judgment on, we believe all of the trials included are sufficiently similar, so there's no issue with transitivity. But If there is, it often kind of presents itself in the form of an inconsistency. So a quick example for transitivity, when we're talking about our loop between treatment A, C and B, if our loop shows that treatment A is better than treatment C is better than treatment B, then treatment B is better than treatment A, it's actually nonsensical, right? We're kind of in this strange loop where, if A is better than C, then, and C is better than B must be worse than A. But if we see something like this, you might wonder, how, that doesn't really make sense. And you'd have to look into the reasons why, and it can be an issue of transitivity. So you can imagine if this is maybe a study in, let's say, hip fracture management, and we find that, treatment A or intervention A, all of these trials are generally in an elderly population, and all of the trials between C and B are in an elderly population. But the general comparison between A and B is often done in younger individuals. Then this is where we have a systematic discrepancy in the overarching kind of eligibility criteria that we've included. Despite the eligibility of the network, it may have been to include any hip fracture patients, but there may be a natural imbalance in who's represented in the trials doing these comparisons, and that's often the nature. And I've made a quick example that's maybe overly obvious, and you might say, well, nobody would ever consider doing this, but I think it illustrates the point. For knee arthritis, we could say we developed this network where we have joint replacement, and there's a few studies that are to conservative care and physiotherapy, and there's a few trials comparing surgical versus conservative treatment. There's also trials of injections to conservative treatment, and there's also trials of NSAIDs to conservative treatment, as well as some between injections and NSAIDs. Now, we don't have any direct evidence. There's no trial out there comparing, pain and function in knee arthritis between surgery or using NSAIDs. But we would have an indirect estimate here that perhaps says NSAIDs provide better functional improvement than surgical, joint replacement. Obviously, with transitivity, we would consider the patients that are typically given NSAIDs in these trials are almost certainly, earlier stage OA. They're a distinct population from the population generally going in for a replacement. So this indirect estimate becomes a little bit nonsensical to make. And this may be an obvious example, but it's something to consider in any network. Are all of the treatments really used, not just broadly in the same population in terms of knee OA or whatever you may be looking at, but really is it logical that all of the treatments are used for the same severity of disease in the same general patient demographics and types? All of those things can pose issues for transitivity. So we have a couple minutes, so I'll go through quickly. Again, it's just some considerations. There's a lot of data in NMAs. There's an opportunity, obviously, to cherry pick or for publications to cherry pick, and this really brings up the importance of having a protocol to ensure that you're explicitly stating everything that you will be presenting and showing. And also, like we've said about transitivity, there's no real rules about what meets transitivity criteria and not. It all comes down to clinical relevance and judgment. And I would argue that that really isn't unique to network meta-analyses, but it's certainly important to consider, talking to, if you're not, a clinician, talking to clinicians on the team to ensure, are these comparisons that we're potentially making through a network logical from, a clinical perspective? Do they make sense? Lastly, we'll go through results presentation. This is, again, that network meta-analysis for antidepressants. And I'll say this is their key table. This is the results they saw. Does anybody have an issue with this? Does anyone want to tell me which antidepressant works best? What the takeaway is? Because I'd say I have no idea. This is incredibly, the benefit of network meta-analysis providing a very large amount of information is almost its downfall as well in how do you present it in any way that someone's going to make sense of. One option that we often see is called ranking, and this is using Sucre scores. It really just lists the treatments from best to worst. There's major issues with this. This is probably the most common way results are reported, but it's oversimplistic, and it really ignores a lot of what we've talked about around clinical importance and clinical relevance of results, and it just ranks them from best to worst. Here's the example from antidepressants, and they've ranked them using these Sucre scores, and a higher score is better. So they've essentially ranked this drug as first, and so on and so forth, using this analysis. But how important is it really to be best without knowing anything about the effect, magnitude, or the certainty around that evidence, the quality of the evidence for that drug? This really boils down to which drug had the largest effect estimate, and really doesn't consider, confidence intervals or anything like that. So it's very problematic in being oversimplistic. So I would caution, focus on Sucre scores, although most NMAs you read probably focus on them. There's a real push to get away from that. Very quickly, this is, a study that we've worked on in promoting better results presentation from NMAs using a table that summarizes benefit, outcomes, and adverse events, and tries to color code them to really show, green is good, red is bad. So the more green you have in benefit outcomes without any red in adverse events, is the most favorable. And obviously something like using opioids, very green, very large effect for pain relief, also very red for adverse events. So this is accepted in BMJ Open, and a framework of kind of how to develop these tables and utilize them. But it's something very important to consider in how you're presenting network meta-analysis results, because you can lose readers and really lose people in general if you present your results in a table like I showed you from the antidepressant study that nobody's ever going to take the time to try to decipher, and it's really going to not have the impact that you may have thought it would, despite how much data and evidence is actually there. Finally, so just some tools for network meta-analysis. The Prisma checklist, they have a checklist, an NMA checklist. Again, if you're conducting an NMA or you're appraising an NMA, that's probably the first place I would go, is look at the checklist, ensure that you've covered everything in that checklist. GRADE also has a network meta-analysis specific aspect of GRADE. Conducting an NMA, I would say if anybody's looking to conduct their own NMA, Stata and R are the two that people most often use. It's a bit less accessible in the sense of, from a pairwise meta-analysis, and Review Manager is fairly intuitive and easy for people to, to conduct those analyses. Networks aren't quite there. There is an online program called Cinema that is fairly intuitive in, you plug in your data and it auto generates everything. But I'd caution people using it in not knowing the details around what's actually happening in the analysis, and using something like Cinema where it's just pumping out the results and you're not certain of how and why certain things were done. So that's one consideration, but there's all kinds of workshops on how to use R, how to use Stata for NMA if anyone's really interested in getting into conducting them. So with that, we have 1 minute to spare. So does anyone have questions? Anybody, general comments, again, about outside of what I've presented? I know it was kind of specific to stats type concepts, but I'm happy to kind of answer any other questions anybody has.
Great, thanks, Mark. Do you want to put your email in the chat, just so, because we're getting tight on time. So if anybody has a question and maybe they would rather email you or reach out to you separately. So there's Mark's email if anybody wants to reach out to Mark with any questions or specifics towards any meta-analyses that the team's working on. That was great, Mark. I really enjoyed it.